Good morning, everyone. Welcome to our second day. I just wanted to say one thing that I forgot to say yesterday. I know some of you know, maybe even all of you, but the Jazz Festival is happening now in the city, and there are lots of free music shows around Place des Arts and other places. So I highly encourage you to just go around. It's also very nice weather today. So I just wanted to let you know that this is happening in case you were not aware. There are also some paid shows that you have to pay to go into venues, but otherwise lots of free shows just outside in the street or in different places. So go take your time and enjoy, because it's great weather today and great music. So with that, I guess we'll start right away, the first session. We do have a small change in the speakers order. We have one speaker, Ari Zimran, who got stuck in the airport. So hopefully we'll make it mid-session and we'll still give the talk at some point. If not, it will be later on during the day. So we'll see how it goes, but we'll start with the second talk, actually. So.
All right. Good morning, everybody. Hopefully you had a good time yesterday and you're ready for a good day of a lot of good talks. So I'm Ignacio Mata. I'm at the Cleveland Clinic. I'm going to chair this session, Genetic Considerations of GBA1 Associated Neurodegeneration. As Zip said, we'll skip the first one. We'll start with Sonza Scholz, who is coming from the NIH, and she's going to talk about GBA frequencies in PD and dementia across different populations. Sonja.
All right, let me just pull up my slides. All right, good morning, everybody, and thank you so much for the opportunity to speak today. So this was a topic that was assigned to me, and so when I realized, there's actually a pretty big body of literature at this stage, I tried to do, the best job in really finding the important studies that highlight differences. If I missed one, please be kind to me. But most of of my points today are really focused on the literature that's coming out of Parkinson's disease. The dementia populations really haven't been well studied, and so this is an important knowledge gap, but I'll try to comment on it as much as I can. So here are my disclosures. I'm at the NIH and I serve on a couple of editorial boards and scientific advisory councils. So the topics I want to discuss this morning is, I want to zoom in on the genetics of the GBA locus. What does the gene look like? What's the locus look like? And why is it just a complex region in the genome that even today with modern technology, sequencing technology, we're still underestimating the kind of allele frequencies that are coming off it. There's a lot of allelic heterogeneity across the various populations, and some of the estimates, especially in older studies, may have underestimated the allele frequencies, and so take it with a pinch of salt. What I say, that some estimates may actually change now that we are learning that the introns are actually important too, but most people haven't actually looked at it. And then I'm going to highlight some important knowledge gaps that we may be able to address as a research community. So let's zoom in on the GBA gene first. So we've already heard this is a gene that is located on chromosome 1. It has 11 exons, and this is already an outdated slide now. So if you look at the literature, it says 5 isoforms last week, and 2 days ago there was a very interesting study by Mina Reiten's group where they did long-read RNA sequencing across GBA and its pseudogene, and they actually found 25 different transcripts coming off, many of which are actually then resulting in protein coding genes that are differently translated across different brain regions. So there's a lot of heterogeneity that may be attributed to those isoforms, and nobody really knows how it actually relates to the phenotypes that we're seeing. But in its canonical form, it forms the typical glucocerebrosidase enzyme, which is 536 amino acids in length. What makes this locus particularly challenging is the fact that there's a pseudogene that is located about 22 kb downstream of the GBA1 locus, which has a very high homology to GBA1. And the problem is that this particular homology makes it very prone to so-called recombination events. And even to this day, sequencing across it can be actually quite challenging. And depending on the studies that have been employed, some of which have only focused in, seems to be jumping on me, focused in either on, exon 9 and 10, where the most common alleles have been located, but even the newer sequencing technologies, due to difficulties with alignment to the reference genome and some misalignment from the pseudogene, have underestimated the allele frequencies that actually exist. And if we really want to capture every mutation, we'd actually have to do long-read sequencing, which is still quite costly if you want to do it at scale. All right, so allelic heterogeneity is quite impressive across this, relatively short region of the genome, about 6.8 kb, that's the GBA locus, and you can see variants all across the entire gene. There's about 658 variants described in the UniProt database. And even if we just zoom in on those that are pathogenic or likely pathogenic, there's about 370 of those described, and we can discuss what constitutes likely pathogenic, etc. But it really illustrates that the pathogenic mutations are really found all across the gene, and if you just focus in on certain exons, you're probably gonna miss some of them. There are different types of mutations that have different effect sizes, mild, moderate, or severe, and that's the topic of a different discussion. But it's also very pleomorphic. We see it either as rare mutations or common variants that can be picked up in GWAS studies nowadays, and different diversity of the types of mutations. So missense mutations, insertion deletions, recombination alleles, and some copy number mutations that have been described in this particular locus. So it's really a complicated genetic locus. And now I'm going to take you on a virtual tour across the world, highlighting interesting studies and some allele frequency differences and important insights that we have gained. And so we'll start out in Europe, then move on to Latin America, African at mixed populations, and then Southeast Asian populations in particular. I'm going to highlight, interesting insights, differences, but also commonalities across these populations. And this is really focusing in on the Parkinson disease literature. And then I'm specifically going to spend a little bit of time just trying to comment on Lewy body dementias, which is really a very understudied disease group, despite the fact that it's actually the second most common form of dementia after Alzheimer's disease in the population over the age of 65. Okay, first and foremost, let's go to Europe. There have been many studies. It's the best study population. The frequency estimate of GBA variants and mutations about 12% in non-Ashkenazi Jewish individuals, but much higher in those of Ashkenazi Jewish ancestry, 18 to 31% of those patients. The most common variants that we see are the E361K, which is very prominent. We actually pick it up on GWAS studies. The L444P mutation, which is a severe mutation, is quite common in Europe. N370S and recombination allele, those are the most common ones that we see in European ancestry individuals. When we focus in on Ashkenazi Jewish Parkinson disease cases, N370S is by far the most common one. 70% of the PD GBA cases actually have this particular change. It is about 3 times as common in Ashkenazi Jewish individuals compared to the mainland Europeans. But the other notable difference is that the L444P mutation is less common. So it's about 6 times more common in general Europeans compared to Ashkenazi Jewish communities. So there are some ancestral differences that we're picking up in this community. Now, continuing our journey to Latin America, the literature is still somewhat limited, and not all studies have really assessed the entire gene. So those are floor estimates, 5 to 10%. 10% is really when we look at the full sequence, and there's some great work from Natchez lab in Colombia. And what's interesting about the Latin American population is that it's quite admixed. We see influences from European, Native American, African ancestry people. So it's an interesting population to study. The most common variants that we're seeing are the L444P mutation, which is actually quite prominent, the N370S, a novel mutation called K198E, and then some other alleles that have been described in the European ancestry population. I want to make 2 points. The L4P mutation is actually quite prominent in Latin America and Hispanic individuals, and it's really one of the major severe disease alleles, and really an interesting target for potential gene therapies because we see it across the globe. So it's a very interesting mutation. And then I mentioned there's a novel mutation, which is the K198E mutation. It's a missense mutation that hasn't been seen anywhere else. It appears to be population specific and actually has been described in a genetically kind of isolated community, PISA community there. It's interesting, it's in about 6% of PD cases. It has a sizable effect size. So it is actually quite common. And so for focused research study, these kind of cohorts will potentially be very interesting. Okay, now we're moving on to Africa, and I must say there's the literature is quite limited. What we know is that focused candidate gene studies have estimated that approximately 5% of PD in North Africa can be attributed to GBA mutations, but now the recent GWAS studies, which included a lot of individuals of sub-Saharan ancestry and more admixed individuals, have found this common intronic variant, which could have been missed in other populations because usually intronic variants are not routinely screened for. This is a variant, and I think we have another talk later, so I'm not going to steal the thunder, where we do see some, evidence for a potential founder in the Yoruban population. Just looking in dbSNP at the allele frequency differences, it really seems to be very highly prevalent in African ancestry individuals. And we see it a little bit coming up in those, of European ancestry, Latin American, where there's some admixture, but predominantly it's really African specific, which makes it an interesting study population as well. All right, moving on to Asia. So frequency estimates vary anywhere from 5 to 10%. Again, there's some methodological differences across the various studies, and I think it's probably closer to 10% when you really look at the full sequencing length sequencing studies. And this is a bit of a busy slide. I just want to highlight some pertinent insights. So about 4.3% of Parkinson's disease cases in Taiwan, 3.2% in Korea, and 9.4% in Japan have been attributed to GBA PD. In particular, when we look at those with early age at onset in Korea, is about 10% of early onset Parkinson's disease actually have GBA mutations, and the particularly common alleles are actually well familiar ones, are the L4P, which is really a global high risk allele, and then the N370S we see again and again. All right, and that brings me to the Lewy body dementia field, and that is very understudied, so it fits on one slide. So most of what we know to date is really based on European ancestry populations. And so we have done whole genome sequencing with a special GBA focused caller that improves the alignment at this locus called Gaussian. And when we sum up all of the variant carriers, mutation carriers, about 13% of cases carry a GBA variant or mutation. The 5 most common variants are those that are very well known in the European ancestry population, E326K, which actually shows up in the GWAS, the T369M also in the GWAS, N370S, the severe L4P, and the recombination allele. What is not actually apparent when you just look at the common variants is that when we compare the Lewy body dementia cohort to the Parkinson's disease cohort, also using the same methodology, whole genome sequencing with the Gaussian alignment, we see an enrichment of severe mutation carriers in Lewy body dementia. So there's probably some genotype-phenotype correlation. So if you carry more severe mutations, you're at higher risk for developing dementia. Not surprisingly, the frequency of Lewy body dementia that can be attributed to GBA is higher in individuals of Ashkenazi Jewish ancestry. Again, we're seeing the known alleles, N370S, but less common, L444P, etc. And then it gets really hard. I've really been scraping through the literature. It's, we can try to extrapolate based on some descriptions of, PD cases with mild cognitive impairment, which probably will progress to Parkinson's disease, but there's really a lack of knowledge and systematic investigations. Also, it's very challenging to diagnose Lewy body dementia, and ancillary diagnostic tools such as the seed aggregation assay are not readily available in our global communities, and so that's a huge unmet need for us. And I think we really need to double down in trying to map out the genetic architecture in the Lewy body dementia population. All right, and so I already mentioned some of the pertinent knowledge gaps. So while we have really made a lot of strides forward in mapping out the genetic architecture in ancestrally diverse population, we still don't understand it very well. And I think there's more work to be done to really identify population-specific alleles, but also alleles that are really commonly shared across not just Parkinson's disease, but also Lewy body dementia. And Lewy body dementia is perhaps the most understudied disease or the form of dementia that exists. And so as a research community, we need to really try to recruit these patients, get them into brain donation programs if it exists, and really try to go straight for long-read sequencing in some of those communities if we can. And sequencing diverse cohorts with full-length sequencing and perhaps even long-read RNA sequencing to identify those transcripts is something that we need to work on as a research community. So it's a bit of a busy summary slide, but what we have learned about population differences is about 12% of PD in Europeans is attributed to GBA. Variants, 5 to 10% in Latin America and Asian communities. And then now 39% in African, but it's, this is because they have looked at intronic variants, and so perhaps these numbers will really change once other studies are really looking at the intronic sequences. We can attribute Parkinson's disease, Lewy body dementia to GBA mutation about 31% of Ashkenazi Jewish individuals. Makes it a really, for a research point of view, it makes it a really high yield population. There are population-specific alleles. I mentioned the Colombian allele and the African variant, the intronic variant. The L444P is a great example of a severe mutation that has been seen all across the globe. We see it in 13% of Lewy body dementia cases of European ancestry. And we need to do more sequencing, and we need to recruit more patients into the field, and also train more people to recognize these very challenging diseases. And so with that, I'm going to just say thank you to all of my various collaborators. Not all of them fit on the page, and my funders, and of course the patients and families who worked with us. And so I'm going to stop here and maybe take a question or two.
Can you say anything about multisystem atrophy and its relationship to this whole
Yes, of
thing?
course. Thank you. That's a great question. So candidate gene studies have suggested that GBA mutations are associated with multiple system atrophy. Having that said, we've just recently done whole genome sequencing of about 900 MSA cases of European ancestry and about 3,000 controls. And we really very aggressively corrected for ancestral differences. And when we do that, although we sometimes see an N370S mutation carrier, we do not actually see a statistically significant enrichment of GBA mutations or variants in the MSA population. So as far as I can tell, it doesn't seem to be playing a major role in multiple system atrophy. It's a great question. Thank you.
I asked it because I'm aware of a family where let's say the grandmother, on the paternal side, had MSA. Her son was diagnosed with fairly early onset PD and developed dementia and passed away. His nephew i.e. his older brother's son, was diagnosed with early onset PD. And the gene mutation was IBS 2+1.
Okay.
So
This is an interesting family.
yeah.
Yeah.
So, and an Ashkenazi family, actually.
Fascinating.
Yeah.
There's another family that I'm aware of from Bavaria, Germany, where there's also a lot of Parkinsonism, Parkinson disease, Lewy body dementia, and one pathology confirmed MSA case.
A similar idea.
So
yeah.
it may not be commonly associated, but perhaps some of the rare variants could play a role. There's also a little bit of literature from Christos Prokakis' work where he showed that sometimes he can, in MSA brains, he can pick up duplications or triplications of the entire synuclein genes in some of the brain regions, but not in all of the cells. We have looked for, duplications, triplications across the brain tissues. We didn't see it, but at least it's not a common risk allele as far as we know in MSA, but
Thanks so much.
somebody else may be interested for follow-up.
Thanks. So, Sonia, that was a nice overview. I'm wondering, it occurred to me when you were describing the alternative splicing at the locus and also with the paper that just came out, has anyone carefully mapped the GBA variants that are known to be associated with PD to the different isoforms? Because if there were some variants that only hit selected isoforms, it's quite possible that some of the penetrance or incomplete penetrance that we see would be explained not necessarily by enzymatic effects, but by the possibility to compensate due to other isoform usage.
Yeah, I mean, that's a great point, and I don't think there's any great studies that have really systematically mapped the transcript levels that are coming off. But I think now with that particular study out, I think that's really something we should look into, and it may really be at the core of some of the clinical heterogeneity that we cannot explain right now. Hi.
Just one gap that I would add is we're talking of variants, and we see that the impact of the variants in the clinic is very, very variable.
Yes.
Should we consider going for haplotypes, knowing that one, for all people, is not so important, but the others might have as well different modified or other haplotypes that might as well explain
Yes.
the clinical variability.
That's a great point. I deliberately didn't do that. Oh yeah, so the question was, there are differences in the effect size that we see even across populations. And, does the genetic background matter? Does the haplotype structure matter? And I think that's a very fair point. I didn't really go into the effect size difference because I know there's a separate talk on that, but it's a fair point. And yes, the genetic background matters. And, we present this disease as like a monogenic variant. That's the only thing that matters, but it's the context that matters and a lot of modifiers that we haven't really mapped out effectively that probably really explains some of the differences. Great question. Thank you so much.